So this presentation will explore the WI38 cell line and its biological concepts, its medical impact, and social relevance. So here's the introduction. The WI38 cell line, it's an extremely important human cell line with many biological and biomedical uses. It's derived from the lung cells of a 3-month-old fetus in 1962. And since its discovery, it's been used in vaccine development and biomedical research. And some examples of that is in research related to cellular aging, cancer research, virology, and etc., like toxology and other things. In this presentation, we'll explore the biological, medical, and social or societal aspects of the WI38 cell line. So first we'll go over the cell division process, which is called mitosis. And the first step in this is interphase. So this is before the cell division actually starts. And this has 3 phases, the G1 phase, the S phase, and the G2 phase. And in these phases, basically the cell just grows and goes through normal function. And then in the G2 phase and S phase, it starts to get ready for replication. And the mitosis actually starts in prophase, which is when the chromatin condenses into chromosomes, the nuclear envelope breaks down, and spindle fibers begin to form. Then there's metaphase, which is when chromosomes line up in the middle of the cell, and then the spindle fibers attach to the centromeres. After that, we have anaphase, which is where sister chromatids are pulled apart to the opposite poles of the cell. And then there's telophase, in which the nuclear envelope reforms, and then the spindle fibers disassemble. And finally, we have cytokinesis, which is when the cell splits into 2 daughter cells, and then that's the whole process of mitosis, and you have 2 brand new daughter cells, which are identical. Oh, and this is a diagram of mitosis right here. It shows all the stages of mitosis. The origin of characteristics of the WI38 cell line. So this was developed by Leonard Hayflick. So these cells, again, were derived from the lung tissue of a 3-month-old female fetus who was aborted. And these are diploid cells with a finite lifespan of around 50 divisions, and this is called the Hayflick limit. These are grown in Eagle's minimum essential medium, and this is a basal culture medium with specific nutrients that allow the cells to grow and be able to survive. And for long-term storage, these are usually stored in really low temperatures in liquid nitrogen usually. So contribution to vaccine development. These viruses are grown in the... So first, viruses are grown in the WI38 cell environment. And since these cells are human-derived, they mimic how the viruses would behave in normal human cells. And the normal human cell's body, and this is what allows it for there to be much more detailed studies about how vaccines and treatments can be formed. And with the help of these cell lines, many vaccines and treatments have been developed. And it's estimated that around 4.5 billion people have been prevented from being infected by diseases, and around 10 million lives have been saved. So examples of these vaccines and treatments that come from the help of the WI38 cell line are that some of these vaccinations were made for rubella, measles, mumps, polio, rabies, hepatitis A, and so many more diseases. This cell line was super important as it was able to create so many vaccines, which saved so many lives. It's been used in toxicology, in which they're screening new drugs and chemicals toxicity in human cells. And the cell line has also been used in gene expression research, which helps us understand how genes are regulated in humans. And this is a chart that shows how many lives have been saved from vaccines from 1974 to 2024, and it is tens of millions of lives. So it's really important that we have the WI38 cell line. So the history of the WI38 cell line, it was developed by Leonard Hayflick in 1962 at the Wistar Institute using lung tissue from a 3-month-old female fetus. And fibroblast cells were isolated and cultivated, and these were created to replace animal-derived cell lines, which would help produce better vaccines and treatments. This cell line also disproved the theory that all cells divide forever. So before this, people thought cells would just keep dividing on, but this was proved wrong, as it showed that it was for a finite amount of time. And it's also used for vaccine development and biomedical research. Ethical concerns. Now, there were some ethical concerns about how these cells were derived. It was a controversial topic. Some people say that it was ethical because it came from a therapeutic abortion, but others argue that it's unethical because the fetus should not have been used for its cells. And many argue that it was extremely important that we had this because it saved tens of millions of lives and has also helped prevent billions of people from being infected from disease. Public health impact. So at the time of the cell development, there were not many ethical laws or regulations for research. The Institutional Review of Boards was formed under the National Research Act of 1974, which helped establish informed consent. And these regulations have been seen by the American Type Culture Collection and Coriell Institute, and they are who document the cell's use in experiments. Personal reflection. The WI38 cell line, it's made a really big contribution to medical science, and it's been a base for all vaccines and new treatments. It's impacted my life personally by having access to all these vaccines, and it will continue to as we keep discovering new things. I personally believe that human-derived cells for medical research purposes are ethical as long as they are safely derived with informed consent. And reflection question would be that would you accept a vaccine if it came from an unethical origin? Conclusion, the WI38 cell is extremely important as vaccines were developed, saved countless of lives, and although there is some debate, it has taught us so much about biology and medicine. And here are some references.